The specific therapy for acute, recurrent acute, and chronic pancreatitis is still lacking. In addition to different environmental factors such as alcohol consumption and smoking, it has been increasingly clear that different genetic variants can predispose or cause chronic pancreatitis, especially in younger patients. The different genetic factors lead to pancreatitis through different pathomechanisms. And by identifying these variants, theoretically allows us to develop a specific treatment for patients with pancreatitis depending on the variant we identified. And it also allows us to develop personalized treatment options for patients. Genetic variants predisposing to pancreatitis lead to the disease to three different pathogenical pathways: the misfolding-dependent pathway, the trypsin-dependent pathway, and the ductal pathway. The most well-known is the trypsin-dependent pathway. The point of this pathomechanism is the early intrapancreatic activation of trypsinogen inside the pancreas that will later will activate more trypsinogen and other digestive enzyme that will eventually cause the disease. The misfolding-dependent pathway is based on genetic mutations that result in misfolding of different proteins that is connected to inflammatory pathways on certain points through ER stress, and that will lead to pancreatitis independently from trypsinogen activation. Genetic testing of human cationic trypsinogen, chymotrypsinogen C, SPINK1, carboxypeptidase A1, and other digestive enzymes, and also different ion channels are generally recommended for patients for pancreatitis. These recommendations, however, do not aim the purpose of genetic counseling, and they do not show us information about the clinical significance of individual variants. Therefore, one aim of our research is to cover this and to determine different clinical significance of different individual variants. To develop therapeutic approaches for pancreatitis, genetic mouse models or genetic animal models that may recapitulate the human phenotype of different mutations are needed. The other aim of our project is to characterize pancreatitis of different genetically modified mouse models. And in order to do that, the so-called T7C140S mouse model we will test in order to characterize the natural cause of pancreatitis, and in order to try out and maybe develop new therapeutic targets for treating this disease.